Okay, our last topic of chapter 1 is scientific approach. So basically, what is science? How do you think about science? Like, how do you think like a scientist? So, how does science work? So, biology, as I kind of mentioned in lab, is the science of life, right? So, we have multiple different types of science, right? We think of chemistry and physics and biochemistry. These are all science, but biology tends to think about how we go about studying about life. And so, life isn't just like looking at like um, on the surface, like how things interact, but also like within us, like how do our cells interact with each other? How does our metabolism work, and so on. And I went into science because I was a very curious person, right? So that's probably one of the reasons why you guys maybe are interested in biology, is because you kind of like a curious person. You want to know how the world works, and so it usually begins with some kind of observation. You make some sort of observation. You make an observation that the sky is blue. You make an observation that like birds eat only certain type of like seeds and so on. And with this, once you make the observation, you will form some sort of hypothesis to try to explain for why that might be the case, right? And so with a hypothesis, you want something that you can actually test. So we want to actually be able to test our hypothesis to either help support or reject whatever our observation is, right? So we're trying to explain something and we want to see, oh, is that the case? Is it true or not? Okay, so let's kind of take a step back and just see what is science. So science came from the word psycher, which just means to know, right? So knowledge and trying to understand knowledge and trying to see, gain this knowledge of how the world works, right? And it's different from the humanities and the arts, and it's distinct from religion because we're not dealing with like spiritual and supernatural. We're dealing with making observation. It's very objective. We have to we form hypotheses. We have to do different experiments to kind of support our hypothesis, and then we form our conclusion that either supports or rejects our hypothesis. And there still remains a degree of uncertainty. So what do I mean by that? That means that once you have supported your hypothesis, it is different in that it's not really a fact, right? Let me move. My face is in the way. Okay, move myself up there. Okay, so there's a level of degree of uncertainty. So an example of that is in biology, for example. You guys are too young for this, but like I remember when I first learned biology, there was we were like, oh, our whole DNA is only so much of our DNA, such a small percentage actually encodes for genes. And so the rest of this DNA like doesn't do anything. We just have all this junk DNA. And we get a lot of our DNA, believe it or not, from viruses. So a lot of our DNA is composed of viruses integrating their DNA into our DNA. And so people are just like, oh, this is just junk. Like we don't actually need all this DNA that's there. Well, it turns out as we explore and we make more observation, we do more scientific experiments, that those junk DNA actually does play a role. So it does play a role in our DNA, and they help regulate the expression of these genes, right? So they're not just there doing nothing. They're not useless like what we used to think. We used to think that they're junk DNA, and now we find out that they're eRNAs, they're long non-coding RNAs, they're microRNAs. There's all these different categories of RNA because they have so many different functions that originally we just didn't realize that was the case. We didn't know that they had all these functions. So that's what I mean when I say there's a level of uncertainty, meaning that we might be able to form these theories, but as we do more experiment and make more observation, we have to kind of adapt to that, right, and evolve with that. Like similar to life, we kind of evolve as we find out more information. Okay, so what is the limitation of science? So again, as I said, there's a level of uncertainty. So we're always trying to make more approaches or make more observation and trying to further understand. And so a lot of times as we find new information, we realize that, okay, well, maybe our original hypothesis, our original observation, that we have to adapt it and change it, right? So an example is this whole little slide right here with this Pluto for planet. So Pluto was originally thought to be one of the planets, and now it's no longer considered a planet. There's a link, I'll put it in the resource so you guys can take a look at it if you're interested. But they redefine what we consider a planet, and it turns out that they don't consider Pluto a planet because it's not it's not by itself as a celestial body in space. There's a lot of other size like planets that are that similar size within that same space. So it so we used to define planet almost pretty much anything that's round, that's kind of orbiting around the sun, and now we realize that that's not enough of a defined definition for what a planet is. So Pluto is now theoretically is now not defined as a planet anymore. And yeah, you can read the article more if you're interested. But that just kind of goes with the idea that, we make observation, we have to reform our original thoughts and our theories to kind of adapt as more information comes in. Okay, so let's kind of think like how do we go about the scientific method? So as I said, we start off with an observation. So we notice something, and we're like, So then we make a hypothesis, and this hypothesis has to be testable. So we have to make a testable explanation of the whatever we observe. So then we devise a test for the hypothesis, right, which is our experiment. And then we carry out our experiment, we collect our results, and now we can analyze our result and decide, okay, does our result support or reject our hypothesis? And depending on what our results are, we might have to adapt and develop new hypotheses. So let's take an example of this. So you come out of the class and you walk over to the parking lot and you start your car because you want to go home. You're starving. You want to get lunch, right? And your car doesn't start. Okay, well, that's your observation. My car didn't start. So now you have to form a hypothesis of why you think your car doesn't start. Okay, is it the gas? No, my gas tank looks full. Maybe it's the battery. My battery might be dead. Okay, so your hypothesis is the car doesn't start because your battery's dead. So let's see, how do we go about testing that? How do we experiment on that? Well, let's put in a new battery and see. And if your new battery starts, then your hypothesis was supported, right? Your car was dead because your battery was dead. And if you put your new battery in and you go, I don't know what sound a car makes if it doesn't start up, but it still doesn't start, then it's like, oh, okay, well, maybe it's not my battery. So then that's your result. So then you analyze your result and you're like, okay, now I have to think of what else is it. Maybe it's the starter. Maybe it's the connection. Like, I don't know too much about cars, but you know what I'm getting at, right? You have to form another hypothesis and take the time to develop the hypothesis so that you can test it out and see if you figure out how the heck to get your car to start so that you can go and pick up some lunch already because you're really hungry, right? You're getting hangry here. Okay, so that's the scientific approach. So remember, you have to make observation, then you develop your hypothesis, and then you test your hypothesis. And if with the test hypothesis, you look at your results and you analyze it and you decide, do I support my hypothesis or do I reject my hypothesis? And depending on what that is, you might have to form another hypothesis, or you might have to readjust your hypothesis and then start over again with another test and then getting the results from that. Okay, whoops, sorry. Okay. So when we do design an experiment, and you guys are going to do experiments in the lab, so kind of think about this as you're doing it, right? So whenever you set up an experiment, you have an experimental group, which is the group which you're going to change something about it, and we'll look at an example in a little bit. Something about the variable is going to change because you want to be able to test how does variable changes within this group. Your control group is identical to the experimental group, but you're not going to apply that same change, right? You're not going to apply that same variable. And the variable is what you're actually going to change in order to kind of test on your two groups. So you have your control group, which is you're not going to change anything. Typically, that's what your control group is. That you don't control anything, or you do change something and you have a very clear expectation, like you know what you expect. And your experimental group is the one that you're experimenting on. So you're gonna do some kind of change in the variable to kind of see whether or not they will change. And then you have your variable self. So let's do an example because that'll make it easier to understand. Okay, so here's an example, right? So we want to see, you guys have seen the plants before, So at this point, you probably recognize it. This is the Ilidia plant, which you guys are using in lab, right? Let me put it down. So let's say you have this plant. You have two jars where you're growing this plant, right? So one jar has water in it, and the other jar, you have water and you have phosphate. Okay, so let's think about this. Here you have these two jars, right? Jar 1 and jar 2. And so what are the variables? Which one's your experimental group and what is your control group? What do you guys think? Okay, well, your variable is probably, because the plants should be the same, right? I mean, yeah, technically they're two different plants, but you expect them to behave in the same way. So you have your two plants. One of them has water, the other one has water and phosphate. So the phosphate is the thing that's different between the two, right? So that's your variable is, does it have water or does it have water and phosphate? So your experimental group is the water and phosphate, right? Because that's the thing that you changed, is you added a phosphate. Your control group is probably your jar that just has water in it. So that's your control group. And then the other one actually has your water and phosphate. And you are using this, right? The experiment, whatever your hypothesis is, let's say you want to see if it increases in growth with phosphate, or does it decrease, like whatever your hypothesis is, you would form, oh, I believe that adding phosphate will increase the photosynthesis of these plants, or however you form your hypothesis. And then you do your experiment, you add the phosphate, and then now you have your control and your experiment, and you're going to see what the difference is between those two jars and the growth. And so some things to consider when we set up an experiment is first the bias and the error. So this is our sampling error. So what does that mean? So we want to make sure that when we set up an experiment that we're representing the whole population, right? So for example, if we have a survey and you only you want to see whether or not like in Silicon Valley that all college students are tech majors. Well, if you only go to the Department of Science and Department of Computer Science, and you're very into math and stuff like that, and you're very limited, you only stay in that quad and you survey those students, well then, yeah, it's gonna say that pretty much, yeah, there's a good percentage is going to tech, right? Because you're skipping all the other quads. What about the quads that does the art and the humanity and the child development and the psychology? Like you're missing, all these other quads and departments and stuff like that because you have that sampling bias. You only took like a small sample, right? So size is a big part, right? Like if you have a very small size of your sample, that can also make it so you have errors. So just take that into consideration, right? So for example, if you were to ask who the greatest puppet is, as in here, and you survey a bunch of people who are like rock and roll drummers, then they're gonna be like, oh, I love Animal. Like Animal was the best one. Like he was the best puppet. They're not gonna go for Miss Piggy or Gonzo or Fonzie because they're biased. They connect with Animal, so they're gonna pick Animal as their choice. So just take that into mind when you are setting your experiment, that you don't end up with a lot of sampling errors. And as we mentioned before, the whole theory is that hypothesis has been tested many times, and we're supporting it, but it has not yet been found incorrect. So then we consider that a theory. But as we mentioned, we can actually change our theory as new information comes in. So keep that in mind. As new information comes in, we have to kind of adapt to that as well, right? So, but the scientific theory is a hypothesis. It's been tested over and over again, and each time the hypothesis kind of comes to the same conclusion, and that's when we would actually go from a hypothesis to an actual scientific theory. Okay, I hope this section of the lecture kind of makes you think a little bit more like how to start thinking like a scientist does. And this is the end for chapter 1. All right, we'll move on to chapter 2 next. Bye, guys.